Hello students, this is Mrs. Kleins with your next section of video notes over passive transport. There are two forms of transport that we will talk about, passive transport and active transport. Passive transport does not require any cellular energy or ATP. Examples of passive transport are diffusion, osmosis, and facilitated diffusion. You can see two of our examples of passive transport on the right side of the slide. This is where solute is moving from a high concentration to a low concentration using no energy. Active transport will require cellular energy or ATP to move substances from an area of low concentration to an area of high concentration. Some examples of active transport are endocytosis, exocytosis, and membrane pumps. Let's first talk about diffusion. Diffusion is the movement of solute particles from high to low concentration. So you can see in this image, you've got your cell membrane, the phospholipid bilayer, with the hydrophilic phosphate heads and the hydrophobic tails facing towards each other. The solute in diffusion can pass through the cell membrane from an area of high concentration to an area of low concentration. It will continue to do that until you reach equilibrium, which is an equal solute concentration on both sides of the membrane. Again, diffusion does not require cellular energy. It can be affected by the concentration of the solute, the temperature of the solution or the membrane itself, the pressure, including air or water pressures, and then the permeability of the membrane, meaning how easily does it allow the movement of substances. So, small nonpolar molecules can more easily diffuse through the cell membrane than large polar molecules. You may notice diffusion in everyday life when you smell cooking, or you notice someone's got on some strong perfume or cologne, or if you have ever made or watched tea being made and diffused through the water. Here is another example that shows food coloring being dropped into the water. You notice initially it starts off very concentrated, and so the solute, which is the food coloring in this case, will diffuse out through the water to remove the concentration gradient or establish a balance across the solution. Here is a graphic that shows a higher concentration of solute or molecules outside of the cell and a lower concentration inside the cell. So that is going to favor the movement of molecules into the cell until you reach equilibrium or an equal concentration of molecules on both sides of the membrane. Here is a graphic that shows a lower concentration of solute or molecules outside of the cell and a higher concentration inside the cell. So that is going to favor the movement of molecules out of the cell until you reach equilibrium, or again, an equal concentration of molecules on both sides of the membrane. Okay, osmosis is similar to diffusion, but it is different because it is the movement of water molecules across the cell membrane. In this case, the solute of the solution is not moving. It is the water itself that moves across the membrane. And what it's going to do is it's going to move towards the area with the higher concentration of solute because that means the water concentration is lower. So you see on the right side of the slide that the water molecules are moving to once again establish equilibrium or to remove the concentration gradient. Again, no ATP is required for osmosis to occur. It can be affected by the same variables as diffusion, including concentration of your solute, temperature, pressure, and the permeability of the membrane. This graphic is showing the movement of water molecules across a selectively permeable membrane to an area of higher concentration of sugar, meaning a lower concentration of water. You can see the water will continue to move across that permeable membrane in an effort to reach equilibrium or osmotic balance. We also see different cells based on their type, demonstrating different characteristics based on the solutions that they are placed in. In animal cells, including human cells, there are three different solution types. One is going to be a hypertonic solution, which means a greater concentration of solute outside of the cell. So what will happen in a hypertonic solution is your free water molecules will leave the cell and that will cause the cell to shrivel or to crenate. A hypotonic solution will show a greater solute concentration inside of the cell. So what happens in this example is the water that is outside the cell will enter the cell in an effort to create osmotic balance. But as the water enters the cell membrane, it doesn't have anywhere to go. So the cell membrane will continue to swell, or in some cases, it will lyse or burst as too much water enters it. And finally, an isotonic solution is the same concentration of solute on both outside and the inside of the cell. So water continues to move across the membrane, but in the same volume, so the cell does not change size. You can see this graphic too, with a hypotonic solution favoring the movement of water molecules into the cell, as the solute concentration is greater inside the cell. Isotonic, you have a cell that is happily exchanging water, but not changing shape or size. And then in a hypertonic solution, the water will leave the cell because there's a greater solute concentration outside. So you see the cell will shrink or shrivel in response. Plant cells are slightly different. As you know, plant cells have an additional cell wall outside of the cell membrane. So in different solutions involving plant cells, an isotonic solution is actually going to create an environment where water enters and leaves the cell, but the cells themselves get kind of a flaccid and start to lose a little bit of structural integrity. The ideal situation for a plant is that you have the cell membrane that pushes out against the cell wall. This is what makes your vegetables crunchy when you eat them. So this is the preferred normal for plants. As you know, if you have ever owned a plant, you give it tap water or rainwater. You don't add any salt to your plant water before you give it to them because you want the solution to be hypotonic. So the plant cell membrane will push against the cell wall, and that creates some pressure and some structure for that cell. This is what we call a turgid structure, and again, normal for plants. In a hypertonic solution, this is like placing it into a salty concentration. You're going to have excess water leaving the plant cell, leaving through the cell membrane, passing through the cell wall, and you notice the cell membrane will plasmolyze or will pull away from the cell wall. So, keys to remember are that isotonic is the solution more ideal for animal cells, and hypotonic is more ideal for plant cells. This right here, upper left, is going to be a cell in an isotonic solution. Okay, looking at animal cells. This is going to be an animal cell in a hypotonic solution, and this is going to be a cell in a hypertonic solution. And you can see here what happens when you put plant cells in tap water. So in a hypotonic solution, right, you can see nice and full pushing against the cell wall. And over here where they're placed in a hypertonic solution. Facilitated diffusion is similar to simple diffusion that we spoke about earlier. But what it can do is it can transport large polar molecules. As I stated earlier, diffusion works better for small, nonpolar molecules, but sometimes large or polar molecules need to be able to pass through the cell membrane. So examples of large or polar molecules could be glucose, salt, amino acids, which are the building blocks of proteins. These substances are still needed by the cell to perform certain functions like making energy in the process of cellular respiration. So in the case of facilitated diffusion, there are proteins that are embedded in the cell membrane that are used to help transport these larger or polar molecules. Again, solutes will continue to move from areas of high concentration to areas of low concentration, and they do not require energy to do so. What this type of diffusion is going to be affected by is concentration and temperature, but it is also limited by the number of transport proteins or channels that are available for these molecules to travel through. You can see this structure where you have a protein channel and your solute traveling from an area of high concentration to low concentration without the requirement of energy or ATP. This graphic here shows facilitated diffusion on the left and simple diffusion on the right, with no protein channel needed, but in both examples, no ATP or energy is required, and the solutes move from an area of high concentration to low concentration in an effort to reach equilibrium.